Welcome back, life science learners. We're going to continue with DNA, and just a recap of what we've looked at in the earlier segment. So we've looked at essentially what the nucleus was, what it contained. We looked at the different types of nucleic acids. In this segment, we're going to focus on the structure of DNA, and we're going to try and understand what was the discovery of DNA and when this happened, and some important people that led to us understanding the structure of DNA today as we know it. So let's get straight into it and try and unpack what is this DNA molecule made up of, and what does it actually mean for us to know what DNA has been in terms of its discovery. So let's get straight into that. So in this segment, we said we're going to focus on the structure of DNA. So I'm going to explain some concepts as we get into this and try and unpack that. So DNA is a polynucleotide. And if we try and break this word up, poly means many, and nucleotide refers to essentially a building block of DNA. So DNA is a molecule that is made up of many smaller building blocks called nucleotides. So poly meaning many, and nucleotides referring to smaller building blocks. So when we look at comparisons to other organic compounds, if you go back to our organic compounds in chemistry of life, we know that we've got carbohydrates, and we say that we've got polysaccharides. And so these are organic compounds that are made up of many saccharides. Now we're looking at a polynucleotide. So it's made up of many smaller building blocks called nucleotides. So essentially, this concept tells us what are the building blocks of DNA called. And these building blocks we refer to as nucleotides. Another term for building block is monomers. So the monomers of DNA are called nucleotides. Let's get back into it. So a polynucleotide is a very large molecule made up of a string of repeating smaller units called nucleotides. And this essentially refers to the monomers. The single smallest building blocks of DNA are called monomers. In this case, they are nucleotides. Each DNA nucleotide consists of 3 parts, and let's try and understand what these are first before we look at a diagrammatic representation of them. It contains 1 deoxyribose sugar molecule, it contains 1 phosphate group, and it contains 1 nitrogen containing base. And so these 3 components make up 1 single DNA nucleotide. Let's try and unpack that in a diagrammatic representation. So as I said, it contains a single sugar molecule. In this case, we often draw it as a pentagon because it is a 5-carbon sugar, and we refer to that as a pentose sugar. Unlike your glucose, which is a hexose sugar, here we've got DNA, which contains a pentose sugar, which is a 5-carbon sugar. It has a phosphate group as well as it has nitrogenous bases. And that is essentially the 3 components that make up a single nucleotide. And as I mentioned, that a nucleotide refers to the simplest building block of DNA. Let's move on. When we look at the structure of these nucleotides, it's important that we recognize that there are 4 possible bases that can form part of a nucleotide. And these nitrogenous bases, which we refer to sometimes as bases, are adenine, and we often use the symbol capital A to represent that. Thymine, we use the letter capital T to represent that. Guanine, capital G, as well as cytosine with a capital C. So let's try and recap that. So these nitrogenous bases that we see in the DNA molecule, which I've represented here, they could be 4 possible nitrogenous bases. And so they are represented either as a T, a G, a C, or even an A. So that's essentially what we refer to as when we talk about the nitrogen bases. However, when we look at this, there are 5 different nitrogenous bases that belong to 2 main groups. Okay? The 2 main groups that we talk about, that these families are the purine bases and the pyrimidine bases. So these 5 different nitrogen bases, which I'm going to illustrate in the next slide. So we've got adenine, thymine, guanine, and cytosine. These, and in addition to that, uracil, belong to 2 major groups. Let's try and look at the next slide. So as I said, one of the families of groups are called the purine groups. And in the purine groups, we'll find that these are double ring structures, as you can see here. These nitrogen bases basically contain 2 rings, and these are essentially found in DNA. So we call them your guanine purine bases, and they're found both in DNA and RNA, and this is often represented as G, and the other individual or member of the purine base family is adenine, and we represent that as A, and this is found both in DNA and RNA nucleotides as well. The other family that we refer to are your pyrimidine bases, and I often try and focus on the Y in pyrimidine, and that often helps me to remember that the 2 that are in this group are cytosine and thymine, and these are found, your C are found in DNA and RNA, and your thymine is found in DNA only. In addition to that, we've got uracil, which is a type of nitrogen base which is found exclusively in RNA. And these pyrimidine bases are your single ring structures. As you would have seen here, these are all composed of a single ring structure. So guys, we get to the next concept of complementary base pairs. Now let's try and understand the context of what complementary base pairing means for DNA. Now the term complementary goes back to even our understanding of how the lock and key mechanism of enzymes work, where you have a specific shape of an enzyme and a substrate that's unique, and these complement each other in a unique way. Likewise, we've got to apply that understanding to the nature of complementary base pairing in DNA. Now, holding that thought that complementary means that there's a unique base pairing between 2 bases, we need to apply that concept to understanding what complementary base pairing is in the context of the structure of DNA. So let's unpack that. So these base pairs of nitrogenous bases always pair up in a similar complementary pattern. This is specific and is referred to as complementary base pairs. I'm going to pause here and I'm going to give you a chance to look at what I mean by this. Essentially, your complementary base pairing refers to the following: that in a base pair, adenine will always pair up with thymine, and likewise, cytosine will pair up only with guanine. Now let's try and understand that in the context of a diagram. Now guys, here's obviously the chemical structure showing you complementary base pairs. What I need you to understand is that, yes, this is important in terms of understanding the complexity behind the chemical structure of adenine, but more especially than that, it's for us to understand that in any complementary base pair which we see here, there's always a purine and a pyrimidine base pair connected together, and you will see that there's a double ring structure here on your adenine and your single ring structure in your thymine. And this base pairing is unique and will only take place between adenine and thymine. And likewise, we see the group pair of cytosine and guanine being combined together in this unique pair, where you have 3 pair bonds that hold them together in this complementary unique base pairing. Right, so let's try and unpack a more complex structure of DNA. Now, I'm going to refer to DNA as being firstly a double helical structure. Right, what does that mean? So on this side of the board, I've indicated to you a twisted helical structure. Let's try and unpack that, unwind that, and illustrate that as a ladder-like molecule. This is what I'm illustrating in this image, and let's try and understand this. So if we step back and if we look at it, DNA is a double helical structure. Helical, as we understand, in terms of this model. It's double because it has 2 strands. On the one end, you see a sugar phosphate backbone. On the other end, you find a sugar phosphate backbone. So this is the one strand that we refer to, and this is the other strand we refer to. And both of these strands are held together by what we refer to as hydrogen bonds in between. So each of these strands are made up of nucleotides. And as I mentioned, that DNA contains these unique building blocks called nucleotides, which has three parts. It's got a phosphate, a sugar molecule, as well as a nitrogen base. And if you look at this model that represents the double helical structure, we see that there is always a base pair between adenine and thymine, or adenine and thymine the other way, and always only between guanine and cytosine, as we see in there. What this model also represents is that there is a unique complementary shape between the adenine and the thymine, which allows only the adenine to fit in with the thymine and not with the cytosine, because these shapes do not complement each other. And so this is more a representation of the complementary shape between an adenine and a thymine. So if we try and unpack this model again, so I'm going to very quickly go back to what the structure of DNA is. DNA is made up of nucleotides. It's a polymer, so it's a polynucleotide, and that essentially refers to, if we're looking at it, made up of many of these nucleotide molecules. It contains complementary base pairs, as we've indicated here, where cytosine and guanine base pair together, or we have adenine and thymine base pairing. It contains a sugar phosphate backbone, which is on either end, and you can see that made up of these phosphate and sugar molecules that are connected to each other. And this is a polymer, meaning it's made up of many of these monomers. So again, a very busy diagram for us to be able to unpack. So let's summarize that with a few key points. So as I mentioned, DNA is a double-stranded polymer made up of nucleotides. Each strand is called a polynucleotide chain. Again, the concept of being made up of many nucleotides. Each polynucleotide chain may contain millions of nucleotide units. So that strand that we looked at contains many nucleotides. It is in the form of a double helix, the shape of which is maintained by the hydrogen bonds between the organic bases. If we go back to this image, I've pointed out that these base pairs are held together by hydrogen bonds, and it is that hydrogen bond that holds the structure together. DNA contains 4 types of nitrogen bases, and these are your adenine, guanine, cytosine, and thymine. Each strand is linked to the other strand by complementary base pairing of your organic bases. As I mentioned, that cytosine will always base pair with guanine, so C and G will always base pair together, and your adenine and thymine will always base pair in that complementary nature together. So that's the structure of DNA. We've looked at the formation of a nucleotide. We've tried to understand what the formation of DNA is, and the next bit that we're going to focus on is the discovery of DNA. Guys, it's important that we understand that DNA wasn't discovered overnight. There was a process, and as with any scientific discovery, there's often pioneers in research that try and find an investigative question and try and dig further into it. And so we all know that Watson and Crick were recognized with coming up with the DNA double helical model structure and received the Nobel Prize for that. However, this took many years of work by many scientists from much earlier on. And so let's try and understand the timeline behind this and what does it mean to be able to put scientific rigor and years of research to come up to what we currently find as the discovery of DNA. So the discovery of DNA started very early in 1875, and it went on till actually 2003 with more significant progress in the recent years around DNA and its application. And so Frederick Miescher was who identified the DNA. Very early, he did some simple experiments to try and identify DNA. This table talks about essentially the timeline, and I'm not going to spend too much of time looking at this, but it's important that there's some really significant experiments that were conducted. So Erwin Chargaff, he played a very important role in determining that there was always equal numbers of adenine and thymine bases, as well as equal numbers of cytosine and guanine bases. We're going to spend some time looking at some key discoveries in the structure of DNA. Notable is Franklin's and Maurice Wilkins' contribution to this. So Franklin was working on crystallizing DNA and X-raying this DNA. So she had perfected the technique of being able to crystallize DNA and X-ray these. And so she was X-raying these and photographing the structure of DNA on photographs that she was collecting. She had the understanding of what DNA composed of, the chemical components, but there was very little understanding of what the structural molecule of this was. So they were trying to busy put together their model of DNA and what it would look like as a complex polynucleotide. So she had subjected a crystallized structure of DNA into through X-ray beams, and that produced a display of diffraction images of light as it passed through. And this was photographed and created as an X-ray image. And these were significant in her contribution to the discovery of DNA that Watson and Crick later on used. Okay. We also know that this was significant to the discovery of DNA. These X-ray beams produced diffraction images, and that was what was important in understanding the scattering patterns of the DNA molecule. And so what she discovered, that DNA was a double-stranded molecule. Later on, she produced an image called image 51, a famous image that was shared with Watson and Crick, and they used this image to put together with their calculations and findings what their model would look like. Francis Crick and James Watson were the significant contributors who were recognized with discovering the structure of DNA. And so they used a process of trial and error with a whole lot of other calculations that they put together, along with the image that I just showed you now. And Watson and Crick were able to assemble a model of what DNA looked like. And they mentioned that DNA was a double-stranded molecule. It contained two strands that were anti-parallel. It was made up of complementary base pairs, and using that information, they put together this image and this model that we see here. And this model was recognized as the most appropriate and correct scientific display of the DNA structure. And these two gentlemen were recognized for their contribution to discovering the DNA molecule with the Nobel Prize. Sadly, Rosalind Franklin had passed on before this Nobel Prize was awarded to Francis Crick and Watson, and only after a few years had gone was she recognized as a significant contributor to this discovery. And so guys, in this session we've looked at the history behind it. It's fascinating to understand what goes into discovering DNA. When we get back, we're going to try and understand some application questions around this. I think you guys have done well. You need a little break before we get into the last segment.